A pleasant day to everyone and welcome to Advanced Topics in Chemistry. My name is Amerigo B. Ramos Jr. from Bulacan Agricultural State College, and I'm here to present my topic, which is something to do with immunology. So my topic is really relevant nowadays, especially we are experiencing this kind of pandemic of the COVID-19. So I think it's really important for us to be familiar with how this immunology is really relevant to all of us in this present day. So now let's start. So before anything else, let's have our learning objectives. Number one is to describe the different concepts of immunology, identify different structures responsible for immunology, and appreciate the importance of immunization and vaccination. So first we have what we call the host microbe or host microbe interactions. We have what we call the non-specific immunity or innate immunity. Now, we have here the first line of defense or what we call the non-specific resistance, which gives rise to physical and chemical defenses. We also have the normal flora. And then the second line of defense, which is also non-specific resistance, which is yung tinatawag nating phagocytosis, inflammation, fever, antimicrobial substances like complement and interferon. And then the third line of defense, which is specific resistance, we have the antibodies, the B cells, and the T cells. First, we have what we call the detect enemy. So once the microbes and foreign bodies enter our body, the body responds to that. And because of that, we have what we call the immobilize or kill the enemy. And then they will send out alarm. Then they will recruit help, make environment unpalatable for enemy to prevent the spread of diseases. And then this process will be repeated. And that has something to do with our immunization. Next, we also have here the toll-like receptors on the macrophages. And we have the three ways to complement this. And then we also have the phagocytosis, the lysis, and also what we call the cytokine release. Also, there is also what we call the response of our body, which results to inflammation and fever. Now we have here the different steps in what we call the phagocytosis. You can see here a phagocyte inside our body, and then the organism is being engulfed into a phagosome, as you can see here in the picture. Then the phagosome fuses with lysosome to produce phagolysosome. And then the organism is killed within the phagolysosome. The digestion and the breakdown of organisms within the phagolysosomes will take place. And finally, the contents of the phagolysosomes will be eliminated by exocytosis. Next, we have the complement system, which is activated in three ways. Number one is the antibody or antigen augments specific defense. Number two is molecules that recognize bacterial sugar polymer or the mannan. And then number three, the random binding to cell surface or the C3b, which will be discussed as we go on with our discussion. Now, as you can see here, we have the structure for the C3b receptor on a phagocyte, just like what I have presented in the previous slide. And then also we have here the membrane attack complex or the lysis, okay? And then we also have here the inflammation. The C3 and the C5a induce changes that contribute to the local vascular permeability and attract phagocytes. What else triggers inflammation? So these are the tissue damage. And what are features of inflammations? What is the inflammatory process? So all of these questions will be answered as we go on with our discussion. Now, as you can see here in the illustration, we have here the inflammation. So say, for example, we have here the skin surface, which will be punctured by a foreign body. and it is near in what we call the arteriole, the venule, and the capillaries. So a normal blood flow in the tissue is as injury occurs. So what will happen next? The microbial products, the microbes, and there will be a tissue damage. As you can see here in the illustration letter B, substances released cause dilation of small blood vessels and increase blood flow in the immediate area. And last, in letter C, phagocytes attach to the endothelial cells and then squeezed between the cells into surrounding tissue. So that will happen if there will be a foreign body that will enter our system or the first line of defense. Now, as you can see here, there will be a formation of pus. So the site of tissue damage and inflammation, and there will be what we call the diapedesis. So what is that? The attraction of phagocytes causes them to move to the site of damage and inflammation. Collections of dead phagocytes and tissue debris make up the pus often found at the site of an active inflammatory response. And this is an example of a white blood cell and then destroying a bacteria. Okay. Next, the movement of phagocytes in the direction of an infection due to the attraction by complement chemicals released by microorganisms and the remnants of damaged cell membranes is a process called A, phagocytosis, B, chemotaxis, C, diapedesis, D, cytoadherence. So this is just a quick quiz. Okay, so the correct answer here is chemotaxis. Now, also after that, we have, aside from the chemotaxis, which is the response of our body if there is the entrance of foreign bodies or foreign materials, we have here fever. Okay, so fever is a hypothalamus controls body temperature. So as we all know, our hypothalamus controls the body temperature, and if it detects foreign bodies, it will trigger the response of our body to make our temperature rise. Now, and also we have here the pyrogens, resets temperature set point. And what are these pyrogens? These pyrogens also coordinate with the cytokines, and this unfavorable for the bacterial replication because there is a high temperature, so they will not survive. And also this is favorable for immune response, and thus it elicits the phagocytosis process and the lymphocytes replication. All right, so we can really say that fever is somehow important for our body. And then another pop quiz, an enzyme found in our tears, saliva, serum, and mucus that degrades the peptidoglycan of the cell wall of the gram-positive bacteria is called A, amylase, B, lysozyme, C, keratinase, D, streptokinase, and E, peptidase. So the answer is lysozyme. As you can see also in the previous slide or the picture, during the phagocytosis. Okay, so the lysozyme. And then here we have the different chemical and physical barriers in our body. So as you can see here, we have the lysozyme in the tears and other secretions in the phagocytes, the removal of inhaled particles in our nose and throat. And then we have here commensal organisms, which usually thrive in this area, which helps us to eliminate foreign bodies. And also we have here mucus to keep the membrane moist, and also cilia that eliminates or screen or filter foreign bodies. And then we'll also have here the skin, which is the physical barrier, the fatty acids, the sweats, and also the commensals. And what are the commensals? These are microorganisms or the natural flora of our body, which is also considered the first line of defense of our body. And also we have acid in stomach. It can be low or high pH that has something to do with the degradation of different microorganisms. And then we have the rapid pH change from stomach to the upper intestine. And also we have here another microorganisms that are living in our gut. and then flushing of the urinary tract, the pH and commensals of the vagina. So these are some of the chemical and physical barriers of the body. Now, what is normal flora? As I have mentioned in the previous slides, we can say here that a normal human has approximately 10 to the 13th power body cells and 10 to the 14th power individual normal flora. So microbes that grow on external and internal surfaces of the body without producing obvious harmful effects. Now, transient microbial flora only occasionally inhabit the body. And also we have your symbiotic relationship. Those are the commensal, the mutualistic, and the parasitic microorganisms in our body, inside and outside. So you can see here in the picture, okay, so these are the sites where the normal flora thrive. So we have the nose, and these are the bacteria that, or some bacteria or fungi that live there. We have the nose, the throat, the large intestine, the mouth, the skin, the vagina, and the urethra. So these are the normal flora of our body, which somehow has benefit and some harm to our body if would not be taken control. Okay. Next is what we call the importance of normal flora. Is it really important for us to have this? So number one, these normal flora prevent the attachment of invading organisms. Next, it produces antimicrobial substances against other microbes that are pathogens or disease-causing organisms. And then also it stimulates immune system or immune response. And then cause the production of cross-reacting antibodies, and it's a significant in nutritional source of vitamins because there are certain bacteria in our body that helps in the synthesis of vitamins. And also we have here the cause dental caries and gum disease. I think it's not one of the benefits, no? Okay. And then how do we acquire microflora? Now, bakit nga ba tayo nagkakaroon yan? So number one is during birth and within the first 12 hours after delivery. So say for example, kapag normal delivery, syempre dadaan tayo dun sa may pinaka-birth canal ng mother. So most probably yung mga natural or microflora doon na transfer kay baby. And then also the breastfeeding versus bottle feeding. So you can see the difference na kung paano natatransfer yung microflora. And then we have contact with people, environment, and food. Also the eruption of teeth and introduction of solid food. So those are some of the sources. And then what leads to the changes in the normal flora? Number one is antibiotic treatment, immunosuppression, and also the diet, and changes in physiology. Say, for example, there is a hormonal imbalance, or example here is the estrogen glycogen effect. Okay. Now, this is an example of what we call the antigen. This is an example of immunoglobulin. So this is a bacterial cell that contains cell surface receptors, or what we call the epitopes. So we have here the antigenic determinants. And these red structures are what we call the antibodies. So there is a specific region, an antigen recognized by an antibody. And then we have here the T cell dependent antigens. So as you can see here, this is an antigen. And then we have here the B cell receptor. So the B cell receptor binds to an antigen, and then the B cell internalizes the antigen. So after that, the antigen fragment presented by the MHC class 1 molecule, this B cell degrades antigen into peptide fragments, then presents them in the groove of the MHC class 2 molecules that reside on the B cell surface. Then what will happen next? If the T cell receptor of an effector T helper cell binds to one of the fragments, then the cytokines are delivered to B cell, initiating the process of clonal expansion. Now, what about the different type of antigen? We have here the T cell independent antigens. So we have here the B cell receptors, as you can see here in the structure. of the B cell. And then we have here the polysaccharide antigen with multiple repeating subunits. So what happened there? There is a response of young children to these antigen is poor. So that is why as we grow older, our T cell independent antigens somehow developed. Okay, so it evolves. So ano nangyayari nga niyan? So nagkakaroon ng clonal selection and expansion of B cell. So, bakit nga ba mahalaga yan for our, yung mga cells na yan? Actually, it's really important for us to have what we call the cellular immunity. Number one, we have cytotoxic T cell. So, it eliminates cells infected with virus. And also, yung mga example niya, yung intracellular parasites. And also, number two, we have the helper T cells. So, anong ginagawa nito? It mediates B cell proliferation and macrophage activation. So, both stimulated by dendritic cells, both produce cytokines that stimulate own proliferation. So, every time na nagkaroon na tayo, na-expose tayo dun sa antibody, sa mga foreign materials, our body will produce this kind of antibody. And every time na ma-expose tayo dun, i-stimulate siya. Okay, and another, as you can see here in the picture, we have particulate and soluble materials, including microbial fragments. So as you can see here, we have here the cells, the dendritic cells. In the tissue, collect particulate and soluble antigen. When a toll-like receptor is engaged, the cell produces co-stimulatory molecules. So these are what examples of molecules with that. Okay, and then we also have here the T cell receptor that will bind to different kinds of antibody molecules. Okay, so en route to the secondary lymphoid organs, the dendritic cells mature to become antigen presenting cells. Peptides from the collected material can be presented by both MHC class 1 and MHC class 2 molecules. Dendritic cells that have engulfed microbial fragments produce co-stimulatory molecules. Naive T cells that recognize antigen present by a dendritic cell that expresses co-stimulatory molecules may become activated, allowing them to proliferate and develop their effector function. So you see, napakahalaga ng ating mga T cell receptors at saka yung mga MHC class molecules because they help us para magkaroon ng activation every time na na-expose tayo dun sa mga foreign bodies. Okay, so these are also example of that. We have here the effector T cytotoxic cells delivers performed cytotoxins to the infected cells. So kapag na-infect na ngayon yung cells, so magkakaroon ng pag-atake hanggang sa masira na yung cells. Okay? And then we also have here the helper T cell activation and interaction. So ito na nga siya. So number one, the B cell receptor binds to an antigen, and then the B cell internalizes the antigen again. And then parang kagaya siya sa previous slides, no, yung proseso ng kung saan na-expose, nagbabind, and then after magbind, magpuproduce ng molecule na naaalala or na nadedetect ng ating body, and then magpuproduce naman ng another effect. Okay? So ito yon. So example niyan kung paano na-activate naman ni helper T cell ang ating macrophages. So macrophage engulfs the material, and then kapag na-engulf na niya, macrophage degrades protein in the phagosome into peptide fragments. So pag na-digest na siya, tapos magiging peptide fragments na lang siya, what will happen next? The peptide fragments from engulfed material are presented by the MHC class 2 molecules. Then, what will happen next? Dahil nagpo-produce, malalapit na ngayon sa kanya si T cell receptor, si helper T cell. So, magkakaroon ng effector T helper cell, recognize a peptide being presented by the macrophage and responds by activating the macrophage. So, diba? It's really amazing na nagaganap ito sa ating body. Now, we have the applications of principles of immunity. So, saan nga ba natin siya nagagamit? So, actually, we have four examples here, or four types. We have the artificial, the natural, the active, and the passive. Now, kapag daw yung mga immunity ay tinawag siya ng example niya is active and natural. So, natural exposure to antigen induces an immune response. Immunity following an attack of measles. Say, for example, di ba pag nagkaroon na tayo ng measles, halos hindi na tayo nagkakaroon nun kasi nga na-expose na tayo. It's natural and active. What about pag passive and natural? So, the transfer of antibodies or cells produced by others. Example, temporary immunity from antibodies of the mother transferred to infant across the placenta or in milk. So, passive siya and it's also natural. What about active and artificial? So the deliberate exposure to an antigen induces an immune response. So say for example, nagpabakuna tayo. So 'di ba, active siya and then artificial immunization of children. What about passive and artificial? The antibodies in immune serum, so may serum that are introduced to the body, injection of rabies, immune globulin after the dog bite. So pag sinabi nating pag active na siya, parang pag may sakit ka na, tsaka mo lang sasaksakan. Kapag naman yung pag sa passive, tsaka mo palang sasaksakan, para may prevention. Okay? Okay, so that's the difference between the artificial active and the passive artificial. What about the principles of immunization? We have yun nga example niya yung natural or artificial, passive or active. So pag passive natural passive siya, example niya yung immunoglobulin across the placenta. And then artificial passive, use of immune serum globulin. So immunoglobulin fraction of donor blood. And then active involves lymphocytes and confers lasting protection due to memory. So memory ito ng cells, okay? And then this table shows some important immunizing agents for humans. So, di ba, marami tayong mga vaccines, tapos ano yung mga diseases na nape-prevent niya. Ayan. Tapos sino yung mga person na dapat makareceive ng vaccines. Say, for example, we have here mumps. So, type of vaccine is attenuated virus. And person who should receive the vaccine, so same as measles. So, sino sila. So, example pa nito, yung rabies, inactivated virus grown in human or rhesus monkey cells. So the person who should receive the vaccine, people exposed to the virus, people at high risk of exposure such as veterinarians and other animal handlers. So ito pa yung mga example niyan. Now, as we go on with our discussion, di-discuss natin yung different types of vaccine. So meron tayo dito sa tetanus, tuberculosis, typhoid fever, varicella, yellow fever, so on and so forth. And also meron pa yung anthrax, diphtheria, itong hepatitis, influenza, measles, and the meningococcal disease. Okay? Now, so sabi ko kanina, what are the different types of vaccines? And ano ba yung vaccines? So vaccines induce artificial active immunity. So preparation of living or inactivated microbe or virus or their components. So say for example, hindi yung buong virus, yung pinaka-fragment lang niya, yung protein lang niya, yung cell surface lang niya. And then, adjuvants helps to induce better response. Okay? Effective vaccines should be safe, few side effects, and lasting protection, and also low cost, stable, and easy to administer, and should induce appropriate specific response. So we have here attenuated immunizing vaccines. So what are those? These are used to modify yung live, ginagamit siya as modified live microbe or virus. And then induce infection and mild disease and solid long-lasting immunity. And some are single dose, pero yun sa ating bago nga, di ba, we have double dose. So single dose can induce immunity. Potential for spread to other people helps to develop herd immunity. So yung aim natin para dun sa ating COVID-19 pandemic, no? Disadvantages may cause disease, cannot use in pregnancy or require refrigeration, yung iba. Okay? And then examples of these yung nga sa measles, mumps, rubella, sabin polio vaccine, vaccinia for smallpox. Now, but ano naman yung other type of vaccine, we have the inactivated immunization. So inactivated by chemical treatment, but still antigenic. Cannot cause infection. Disadvantages: require several boosters, may cause side effects. And whole agent use inactivated bacteria or virus. Example, I saw polio virus vaccine, then diphtheria and tetanus toxoids. Sa mga baby usual yun. Now, inactivated immunization, subunit vaccines. So use isolated antigens or antigen fragments, a subunit of the total agent. So we have the bacterial toxin o yung toxoid nga, then the protein subunit, the polysaccharide, also vaccine against meningococci, pneumococci, pertussis, and the H influenza. Then the recombinant vaccine, the hepatitis B, require several doses. And that would be all. Thank you very much for listening. So if you have questions, just you can send your personal message to me, right? So thank you very much for listening. Stay safe and God bless us all. Bye.